Okay.
Okay. So I would say, first of all, shalom and hello to everybody. We have a lot of people here from all around the world. I want to thank all the great speakers that have joined us. For me, it's a dream come true to be with the three speakers, including Dr. Brian Drucker, who is always been our hero, and we'll hear it later in the presentation. So I'm really excited and nervous. And before we start the presentation of the great speakers, I would like to thank all of you for joining. And first of all, I want to dedicate this webinar, special webinar, which celebrates 20 years from opening of the IRIS trial, which was the first trial that imatinib or STI or Gleevec, call it whatever you want, was approved for treatment of CML patients. And it's also a personal story for me for 20 years. I want to dedicate it to all the CML warriors that we have lost along the journey in the last 20 years, and especially to two close friends of mine. The first one was a patient of Dr. Brian Drucker, and that's Zavi Miller from Canada. He was a very close friend of mine. They used to call him and me the drug smugglers because we used to send drugs all around the world. We collected from patients drugs they didn't need, and we send, if there are few patients in Iran who like Israel, that's because they received some imatinib from us many years ago when it was not available. And he was also the Zero Club establisher, and that's every CML patient who reached a zero or close to zero, he gave him a number, and that's really gave a lot of hope for many patients at that time. 20 years ago, it was not clear like today, and I'm really proud to be number 50 on the Zavi's Zero Club. And then the second one is Israeli CML patient. He was a member of our board. He was an accountant. He was in charge of the auditing of our patient organization. And he actually tried all TKIs which were available, and he was either resistant or intolerant to all 5 of them. And he, at the end, went through a bone marrow transplant, which extended his life for 5 more years. And unfortunately, he passed away last year, and we lost him. His name is Menachem Kazanovsky. So they are very good friends of mine, but they only represent a lot of the patients from all around the world. So without further ado, I want to start the webinar and the presentation, and I invite Professor Pia Ranani to open this meeting and tell us what was CML like before 2000. What was the story of CML patients before we had the big change started. Professor Pia Ranani is the head of the hematology department in Bellinson or Rabin Hospital in Petah Tikva near Tel Aviv. It's a very big center. She's also the leader of the Israeli Hematologist CML Working Group, and she has been the medical advisor of our local CML patient organization from the beginning, almost 19 years ago. So it's always a pleasure having you with us, Professor Ranani, and the stage is yours. Thank you.
Thank you, Giora, and thank you very much for giving me this kind opportunity to share this day with you and with these great speakers. So good evening, everybody. During the next 10 minutes, I will take you through the journey of CML during the decades between 1960 and 2000. Alongside this universal journey, I will tell you about my personal own CML journey in the pre-imatinib era. In 1960, Peter Noel and David Hungerford discovered and described the Philadelphia chromosome. This was a real quantum leap of an abnormal small chromosome 22, which turned out to be the most significant milestone in the journey of CML between 1960 and 2000. Here you can see the two scientists, Noel and Hungerford, with their publication in the medical journal Science in 1960. In this slide, you can see the major milestones in the unraveling of the biology of CML and its treatment. In 1960, the Philadelphia chromosome, a minute 22 chromosome, was discovered, as I said before, by Noel and Hungerford. During that period, CML patients were treated with chemotherapy pills like busulfan and hydroxyurea. In 1973, Janet Rowley found that the Philadelphia chromosome is a result of a translocation between chromosomes 9 and 22. That's why we celebrate today the CML Patients Day. While in the 80s, the molecular basis of the BCR-ABL fusion gene was established, and treatment with interferon and bone marrow transplantation was introduced. During the 90s, the biological meaning of the fusion gene as a causative factor for CML was established, and the first clinical trials with tyrosine kinase inhibitors were initiated, culminating in the Irish phase 3 clinical trial that set the stage for the fast-track approval by the FDA in 2001 of imatinib as first-line treatment for CML. In this slide, you can see the historic development of CML therapy. As you can see, between the 60s and the turn of the century, treatment moved from chemotherapy pills like busulfan and hydroxyurea to bone marrow transplantation and very high doses of interferon alpha. Patients were bedridden either due to the very high doses of interferon or post-bone marrow transplantation. Both were associated with a relatively high rate of mortality and morbidity. To summarize the main differences between CML before and after 2000, I will highlight the following issues. Before 2000, CML was a fatal disease with poor prognosis, while currently it is regarded as indolent with excellent prognosis. While interferon improved median survival from 3 to 6 years to nearly 7 years, nowadays patients with CML have the potential to survive for decades. Imatinib has replaced interferon and allogeneic stem cell transplantation as frontline treatment, and in the past there were very few second-line treatment options available, while now more powerful tyrosine kinase inhibitors can overcome resistance to imatinib. In this slide, you can see data taken from the MD Anderson Cancer Center database. You can see here the survival curves for CML patients over the decades between 1965 and post-2000. One can easily see the improvement in the 5 and 10-year overall survival over the years, rising from less than 3 years median survival in the 60s and 70s to 7 years and more in the 80s and 90s, and nearly normal overall survival of patients beyond 2000 with the growing use of tyrosine kinase inhibitors. In the time left for me, I will share with you my own personal journey with CML. Like the Philadelphia chromosome, I was also born in 1960, but not in Philadelphia, but in Tel Aviv. I went to medical school in the 80s during the years that BCR-ABL chimeric gene was discovered. In 1992, while being a resident in internal medicine, I published my fourth scientific paper entitled Life-Threatening Hypophosphatemia in a Patient with Philadelphia Chromosome-Positive CML in Acute Blastic Crisis. At that time, treatment for CML consisted of interferon and bone marrow transplantation with a median survival of 7 years. I will never forget my CML patients whose quality of life was very low, many of them being bedridden, depressed, with interferon and bone marrow transplantation. In those years, CML was considered a very frustrating illness to treat for a young doctor as myself in those days. In 1995, I was sent for a postgraduate course at the Hammersmith Hospital in London. This was a turning point in my career. This course was moderated by Professor John Goldman, who was the guru of CML and the mentor of many future leading figures in the field. After that summer course, I knew that this is what I want to practice in, though in those days, CML was not a very attractive disease for a young physician launching on her medical career. In January 1996, Professor John Goldman and his group came along with many other rising stars in the field of CML to a CML meeting organized in Jerusalem by the late Professor Rachmilevich. Among them was a young oncologist from Oregon, Dr. Brian Drucker, who presented his early results with STI 571, later called imatinib or Gleevec. And here you can see his name and the title of his talk in 1996. During that meeting, Professor Goldman also introduced me to Nick Ross, who became my direct supervisor in the MRD lab at Hammersmith between 1996 and 1997. The clinical rounds in the CML department at Hammersmith during those days were rather depressing, but the good place in the West End in London compensated for this. Here you can see Professor Goldman, Nick Ross, and myself. During the following years, I was treating CML patients with either high doses of interferon or bone marrow transplantation, facing the dilemma whether to offer them the single curative treatment at that time, namely bone marrow transplantation, with a chance of 30% mortality, or continue with high doses of interferon, which could offer them a median survival of 7 years. In 1999, I participated in the 41st ASH annual meeting in New Orleans. I will never forget this cold winter day when we listened to the talk given by young oncologist Dr. Brian Drucker about the new drug, STI 571. We all had goosebumps when we left the hall. We felt that it was a real revolution in the treatment of CML and an important milestone in the treatment of cancer in general. In 2001, STI 571, later called imatinib, was approved by the FDA for the treatment of CML first line. 2 years later, I joined the CML support group headed by Giorra, and since then, our paths have never parted. I would like to conclude on a personal note. I started my career as a resident in internal medicine in 1988. One of my patients, named YZ, was treated with hydroxyurea for CML and developed blastic crisis manifested by hypophosphatemia. This was my 4th medical publication. I was then lucky or unlucky to treat patients with interferon at very high doses with a median survival of 7 years. Quality of life of these patients was very poor. CML patients' fate changed dramatically in 2001 with the introduction of imatinib into clinical practice. During the next decades, CML patients went through a fascinating journey with a changing image of CML from a fatal disease into a potentially curable one. I feel I was lucky to be part of this journey, which is still ongoing, and I'm lucky to share my personal CML journey with you today. So I wish you a very nice and interesting evening. Thank you very much.
Thank you very much, Professor Pia Vanani. It really was a nice description what it was like to have CML in the before 2000. And now I'd like to share with you just a short story of myself, CML, and actually my story is reflecting the revolution that happened in the CML in the last 20 years. So I was diagnosed with CML, which was a big shock for me. I was 48 years old in February 2000. It was just after the millennium has changed, and I was sure that the computer has made a mistake. If you remember all the concern we had about computers changing from year 1999 to 2000. But unfortunately, it was not. I went to 3 experts in Israel, top experts. Each one of them gave me a different treatment plan, and I was lost. And I started searching the internet and tried to find my way. What do I want to do? I found a first chat group of CML patients in the beginning of 2000. And then we joined, those who remember at that year, Susan McNamara. She was a CML patient. I think she was also a patient of Dr. Brian Drucker, if I'm not mistaken. And STI was being developed by Novartis, but all the the people who are involved in developing this were telling the CEO of Novartis, Dan Vazela, that it's not worth to develop it. There are too few patients, it's not worth economically, and they should not spend so much money. And there were hesitation whether to develop further this drug or not. And I'm sure that we'll hear much more about this from Professor Brian Drucker. And then we wrote a petition to Dan Vazela, the CEO of Novartis. Within few days, we raised 4,000 signatures. The petition came to his table. There were also many people who came to him and asked to continue. And he decided at the end, against all the recommendation, to develop the drug. And that was the big change. You can read all this in the book he wrote later, which is called The Golden Bullet. I was waiting to join the IRIS trial to open, and actually I was in touch with Professor Brian Drucker. I already made all my plans to go to Portland, to Oregon. I spoke with a local rabbi. I organized school for my 4 children to make sure they can go, and I was planning to go there for about a year. Unfortunately, the trial did not open in Portland, Oregon, and I ended up through the internet finding the IRIS trial opening in Germany, and I was lucky to join a trial with another great doctor, Professor Hochhaus from Germany, who is still my doctor until these days. I was randomized to the interferon and RSC, always asking myself, why do I have to fly to Germany to get interferon when I can get it in Israel free? But after 7 months, I was switched to the STI 571 on 14th of February 2001, and that actually became my second birthday. Imatinib or STI worked on me amazingly. I became PCR negative within 9 months, and I kept the deep molecular response for 13 years. And in June 2014, with the pressure of my doctor, I stopped imatinib to see if I'm cured. And now I'm more than 6 years on TFR with my deep molecular response MR5 kept in all months in all the PCRs that I'm doing every few months. And because of all the hesitation I had and all the lack of information, I decided to use my experience and help other CML patients, both in Israel and also globally. So the skies, of course, of CML have changed from very cloudy to blue, but we all know that they are not completely blue, and there are still clouds in the sky when we talk about CML. There are still challenges for CML patients today. I just list few of them. One of them is a non-adherence of CML patients, which is not clear why a patient should not take a drug which is saving his life, but still we know that about 30% of patients are not taking the drug as prescribed by his doctor. We know about resistance and relapses that we don't always have the solution to, but it's an issue for some CML patients. A lack of information, for example, pregnancy, how do you treat a young female CML patient who wants to become pregnant? It's not always clear, and not all doctors have a consensus about how to do it. In some countries, there are issues about generics and suboptimal treatment. Quality of life is a big issue for CML patients, and we hear it every day. I hear it from my patients, and many of them suffer, some of them more, some of them less, but it's an issue that we have not yet found a solution. And last but not least, access to treatment. Most of us come from Western countries, North America, Europe, and others, where we have all the TKIs available, or most of the TKIs available, and patients are receiving them and living with the disease. But as part of the leadership of the CML Advocate Network, we are very aware that there are so many countries in the world where there is no access to, sometimes not to any TKI, sometimes only to one TKI with no second line, and CML patients are still dying in too many countries in the world, and that's an issue that we are working on. So what are we doing both nationally and also globally to tackle these challenges? First of all, we are doing research to understand the patient perspective and what the patient thinks. I was lucky enough to lead a team of CML advocates to do two big studies. The first one a couple of years ago about adherence or non-adherence with more than 2,500 patients from 63 countries all around the world. And the last one was last year about TFR, patient full stop, to understand the patient's feeling and perspective, what happens to them when they consider. Both of these studies were published in a scientific magazine. The last one we are very proud about was published in Leukemia only in May this year. We participate in advisory board with companies to improve access. Actually, this happens all the time, and we just had a recent advisory board trying to see how we can help the access in the Philippines, and we have a few other countries on our list, and we work with the companies and mainly with the help of the Max Foundation and Pat Garcia Gonzalez, the CEO, how we can improve access for patients in these countries. We are today participating in new drug development as members of investigators, committees, and we work together closely with the leading CML hematologist groups like the ICMLF or the EICML. Professor Salio, I still remember very well the last meeting I participated in Turin when you hosted the EICML meeting a couple of years ago. We are doing educational empowerment for patients to teach them about the different aspects of CML, and I have just released today a new TFR short video which explains to the patients what TFR is and make it in a very visual, easy to understand concept. And most important, we are keeping the hope of the dreams of the patients. And yes, I myself still have a dream. And my dream is to organize a similar webinar like we have today in 5 years when we all will be able to discuss the cure for CML patients. So I thank all my Israeli patients and also the global CML advocates community for giving me the strength and energy to continue my dream. Thank you very much. So now I would like to invite my hero. I don't know if you remember, Brian, the letter I wrote to you, I think 18 years ago, when we organized a book for you, and many patients wrote an album with pictures, and each one shared his own story, and we sent it to you as a thank you for actually saving our life. So I'm really proud, and I think this is for the first time, to host Professor Brian Drucker from Portland, Oregon. He is the director of the OHSU Knight Cancer Institute. And what can I say about him? We have a Facebook group which is called Brian Drucker is the Man. And it has hundreds of CML patients in that group. And really, I'm really excited and honored to have you with us and to listen to your story, how you have developed the drugs that saved the life of so many CML patients all across the world. Please, Brian.
Thank you, Giora, for that very kind introduction, and congratulations on your 20 years with CML and 6 years off treatment. It's a great pleasure to be with you today, and I'd like to start by wishing all of you a very happy new year, Shana Tova, and I wish you all the best for another healthy year. My journey in developing the first targeted therapy for cancer began as a medical student nearly 40 years ago, when I learned about acute leukemia in children. We learned in the 1950s this was a universally fatal leukemia, often within 6 weeks or less. And articles written about this leukemia all started by noting the best that could be done was to try to ease the horrible pain experienced by the children with this leukemia. Over the next 2 decades, this leukemia became routinely curable with combinations of chemotherapy drugs given over 2 years. As remarkable as that was, all I could think about was there had to be a better way. And in my final term paper, I wrote that only through understanding of what drove the growth of cancer could we develop better therapies. And so my journey began. Of course, I still had 3 years of medical school left, then 3 years of residency training, followed by 3 years of training in medical oncology. During my oncology training, I grew tired of giving patients chemotherapy. It rarely worked and was always toxic. So I returned to my long-held desire to do something different and began working in the laboratory of Dr. Thomas Roberts at Harvard's Dana-Farber Cancer Institute to study how cell growth is regulated. I thought there had to be a better way, that we could kill cancer cells without harming normal cells. During the next 7 years that I was working in the laboratory, many strides were made. As you heard from Professor Anani, a few threads in CML had come together. From Peter Knoll and David Hungerford's discovery of the Philadelphia chromosome in patients with CML in 1960, to Janet Rowley's discovery that it was 2 chromosomes, 9 and 22, exchanging segments to produce this abnormal chromosome, to Rick Van Etten and George Daly showing in 1990 that the product of the Philadelphia chromosome, BCR-ABL, caused leukemia in mice. To me, this meant that BCR-ABL caused CML, but most in the field remained skeptical that it was that simple. In the meantime, Alexander Levitsky, working at the Hebrew University of Jerusalem, published an eye-opening paper in 1988 showing it was possible to specifically inhibit the activity of enzymes like BCR-ABL that regulate growth. Prior to that, most people thought that targeting this family of enzymes would simply not be possible. During this time, I had forged a collaboration with Nick Leiden at Novartis, one of the few companies working to develop drugs to inhibit this family of enzymes to which BCR-ABL belongs. Nick had sought my advice as I developed one of the most widely used tools to study the regulation of cell growth by this family of enzymes. And in addition to being a researcher, I was also a doctor, treating patients who might benefit from drugs such as these. Even though at the time I wasn't working on CML, in 1988, I told Nick that CML would be the best cancer to test targeting this family of enzymes. Then in 1990, I took my own advice and began working on CML with the guidance and mentorship of Jim Griffin. I was beginning to make what I thought were significant strides in my CML research. I had published several papers, mostly as a collaborator. I was feeling pretty good about my work. So I met with the man running Dana-Farber to ask for some lab space and a promotion. He sat behind his desk thumbing through my papers. He told me I didn't have enough independently authored papers and I didn't have a future at Dana-Farber Cancer Institute. But I had a backup. I'd been negotiating with the chief of medicine across the street at Beth Israel Hospital. When I went back to close the deal, I was told the job had been given to someone else with more promise. I was devastated. 2 of the most prominent leaders in the field of medicine had told me I wasn't worth the investment. But as I look back, this was a turning point. It was like someone knocked me in the head. I'd gone into cancer research to find a better way to treat cancer. I was tired of giving chemotherapy and had promised my patients I would go into the lab until I found a better way to treat cancer. These events awakened and invigorated my desire to make a difference. It was an opportune time. I believed targeted therapies were the future. Those setbacks gave me focus. I just needed lab space and a fresh start. I accepted a job at Oregon Health and Science University and moved to Portland, Oregon. I had one goal. I wanted to find a drug that worked in my CML disease models and get that drug into the clinic for my patients. I was fortunate to find a wonderful boss in Oregon, Grover Bagby. He said sometimes the best thing you can do for a researcher is to give them an opportunity and leave them alone. And he did just that for me. Within 6 weeks of arriving in Oregon, Nick Leiden sent me some compounds that his group had developed that he thought might inhibit BCR-ABL. I started working with these compounds, one of which killed leukemia cells without harming normal cells. I had exactly what I'd been looking for. This compound would later become the drug Gleevec. But at the time, there were huge hurdles. The drug company didn't think it was worth the investment. They thought the drug wouldn't work, or it would be too toxic. And worst of all, they thought it would never make enough money to warrant the investment in its development. Nor were they willing to take a chance on an unheard investigator from Oregon. So began a 5-year crusade to get this drug to patients. Fortunately, I had a few scientific advocates. Professor John Goldman was the first to recognize the potential of this drug. You heard Professor Agmilowicz invited me to give a presentation in Jerusalem in 1996, the first international presentation of my data. And shortly after that, Moshe Talpaz joined us. And though I felt abandoned by some people I once respected, there was one group of people who never doubted me, my patients. I'm always amazed at how patients believe in you, even when you're just starting out. Bud Romine was one of those patients who believed in me. Bud was a newly retired railroad engineer. He had been diagnosed with CML, and at age 65, he had few options. He tried interferon, but it didn't work and left him with little energy. He was too old for a transplant. His wife of 42 years, Yvonne, a retired bank clerk, kept a meticulous folder filled with charts of his blood counts. The lines fluctuated dramatically from 15,000 to 150,000. She drew another line at 10,000 for the top of the normal range, something Bud hadn't seen in years. But for Bud, the numbers didn't mean much. He only knew he didn't have any energy. He couldn't play golf, and he often felt too weak just to get out of bed. Bud read a story in the newspaper about our laboratory studies. He contacted me and put his life in my hands. Bud knew the average life expectancy for a patient with CML was about 5 years. And with 4 years on his clock, he knew his time was running out. Bud put his faith in me and trusted that I would be able to help him. That faith bolsters you. It emboldened me to take on the biggest drug company in the world and get this drug to them. We had tested the drug in the lab and were pretty sure it was safe. But until you give it to patients, you don't really know. Bud was incredibly courageous. He stepped forward and wanted to be the first patient to be given this drug. In June 1998, Bud's wish was fulfilled, and he was the very first patient treated on our clinical trial. Within 6 months, every single one of our patients had their blood counts returned to normal. Over the next several years, patients traveled from around the world to enroll in our clinical trials, including 2 from Israel. As you heard from Giorgio, patients lobbied for faster access to Gleevec in clinical trials, and this helped spur the drug to FDA approval in record time in 2001. For Bud, the numbers, the approval of the data didn't give you the real picture. For Bud, it meant he could play golf, buy a new truck, and spend his winters with his family in California. Now, almost 20 years later, this previously fatal leukemia has a 95% 5-year survival. And we can tell patients diagnosed with this leukemia that the life expectancy is the same as anyone else their age. And for some patients, as you have heard, we have been able to stop treatment safely. But we know that Gleevec isn't perfect. Some people's leukemias become resistant to this drug and their leukemia returns. We learned an enormous amount about what causes resistance and now have 4 additional drugs targeting BCR-ABL and several more in clinical trials that target the main causes of resistance. Other people struggle with side effects, and we continue to strive to find new ways to manage these side effects and to find more reliable ways to get our patients to a point where treatment can safely be discontinued. Our journey with this leukemia is not over, but the current ability to treat it is so vastly improved that we now consider CML a manageable condition as opposed to a life-threatening illness. I owe the greatest debt to my patients who put their faith in me as we've gone on this journey to find better cancer treatments. I've learned the importance of listening to your patients. Some of the best lessons come from them. Years ago, I said the greatest lesson that they have taught me is the greatest gift in life is hope. But now, 20 years later, I've learned that the greatest gift I've been given is seeing what my patients can do with the gift of life. And I just want to share with you what some of those patients are doing. I'll start with this patient on the upper left, the very first patient, the longest patient continuous on treatment. She's doing things she's enjoying, like gardening. On the upper right, this patient, the first patient treated from Italy in our clinical trials, doing what he enjoys doing, which is dancing. This remarkable young woman who was diagnosed at age 6, almost 20 years ago. She's now a nurse at our children's hospital, and within a couple of months is expecting her very first child. This very close friend of mine who was diagnosed 15 years ago, when he met with me for the first time, he said, all I want to do is to live long enough to walk my daughter down the aisle. He was able to do that a few years ago, and now is the proud parent of is the proud grandfather of his first grandchild. This remarkable woman, one of the very first patients treated from Australia who traveled to Portland in the year 2000. She then returned to Australia and was selected to be one of the torchbearers for the Olympic Games for the Sydney Olympics in the year 2000. These stories are your stories, and it all begins to add up to lots and lots of people who are now thriving, surviving, despite a diagnosis of chronic myeloid leukemia. And for me, this is the greatest reward. And for that, I thank you very, very much.
Well, Professor Drucker, what can I say? I hope the camera is not showing the tears on the side of my eyes, because it was a really, really moving story. I don't know if you know, if I ever told you when we met a few times at Ashes, that actually the idea to start the Israeli CML patient organization came when I supported the CML patient in my city, in Netanya, Dorothy. I think you remember her, Dot. She was progressing from a chronic to accelerated phase, and there was no solution. And we supported her to go to you and receive STI 571 on compassionate program or early access program, not in a clinical trial. And really, you have saved her life. And if you remember at the beginning of the trials where you had the first patients taking STI 571, you used to gather all the patients once a month in the clinic and talk to them about this new drug. How is it working? What are the effects? What are the side effects? And you had a patient called Judy Oram that was writing a magazine about each meeting. And when Dorothy came back to Israel after almost a year, she stayed with you. She asked me to start organizing meetings like the meetings you used to organize in your hospital for the Israeli CML patient. And that's how our Israeli CML patient was born, with 7 CML patients in the small hospital in Netanya. And now we have more than 500 CML patients from all over the country. And it's really, the beginning was due to your meeting. So I thank you for that also. So I want now to move to another great friend of mine, Professor Salio, Joseph Salio, always easier for me to say Beppe, so excuse me if it's difficult to pronounce Giuseppe. We met many times in many meetings, and you presented in many of our CML Horizons meeting for the CML Advocate Network, and it's always great pleasure having you with us. We heard what was the situation of CML before 2000, and how this has changed dramatically in 2000 with the introduction of STI 571 and imatinib. But I think no one of us could have dreamed at that time that there will be one day that maybe we'll be able even to stop the treatment, because we all thought that this is a life-threatening disease where we will have to take a pill or few pills every day for the rest of our life. But then came the first trials of stopping imatinib stim, and I would like you to tell us about this idea of stopping treatment and how did it come to life, and all the experience and knowledge that you have about the TFR. Remember, we did a video together when I interviewed you about this TFR story just a couple of years ago. So Professor Salio is a professor of internal medicine and hematology at the University of Turin in Italy. He's one of the world's leading CML experts, and as I said, I heard him present in many meetings, and it's always a pleasure having you with us. So the stage is yours, and thank you for joining us very much.
Thank you very much, Giorgio. And first of all, let me thank you for the invitation to participate in this very important day for CML patients, because this is just to speak with persons like Brian. I think that is always an emotion. I would say that I was present in Jerusalem, Brian, when you first presented your data in 1996, as you remembered, in the house of in a hotel near the house of Eliezer Rakmilevitz. And indeed, already at that point, everyone was really impressed about the possibility opened by this new drug at that time, the imatinib. But as I would like to remember now, if imatinib and all the, let's say, the second and third generation TKIs were able to control the disease and give to the patients the possibility of an overall survival similar to that of a control population without leukemia, our goal now is becoming just to achieve a real cure. And we call this status TFR, treatment for remission, which is operational cure according to the definition given by John Goldman some years ago, which means that the patients may live without showing a relapse, a recurrence of the disease, even without taking the TKI. So this is something that after a certain number of years becomes a real cure. As you know, the story started approximately 10 years ago when Francois Xavier Mahon published the first paper concerning this trial, the STEAM trial. And as you can see, what was surprising, that after an initial period, after the discontinuation of the therapy, that almost a huge number of patients, huge 60% of the patients, indeed show the recurrence of the disease. Indeed, there was a plateauing of the remaining cases who were able to persist in a deep molecular response without taking the TKI anymore. So this was the first scientific demonstration. There were already anecdotal reports, but this is the first trial really introducing the concept of TFR in the story of CML. And show us that indeed treatment discontinuation is possible. We didn't know how many patients could achieve this goal at the time, which was the denominator, let's say, of the CML patients which were achieving this goal, but at least the concept was established. After that study, many other studies, and this is almost an old slides because it was just 1 year old, more than 50, but so far I think more than 60 or 70 discontinuation studies have been published. involving more than 4,000, probably more than 5,000 nowadays patients, and no more of 3-4 cases of progression have been reported so far. We are lacking precisely this information. We are trying to establish now a precise registry and to compare the number of progression which occur in patients who are discontinuing the therapy with respect to those who are continuing the therapy. Of course, similar categories of patients, patients in deep molecular response. But for the moment, what we know that apparently the treatment discontinuation, TFR, can be considered safe. So it's not dangerous to try to discontinue the therapy. And then a very interesting phenomenon that was explored some years ago by the group of Kim in Korea, as well as more recently even in Australia, you may see that even after stopping the therapy, some of the patients who are still scoring positive using very sensitive PCR, indeed they show a progressive decrease in 1, 2, 3, 4 years of the residual leukemic cells. This is based on RNA, so on the expression of the PCR fusion RNA. But the same experiments was more recently repeated by the Australian group, as I told you. They did a DNA showing that indeed the leukemic cells not only are in some cases expressing, but also their presence is progressively decreasing until a total exhaustion in most of the patients. These are their experiments. So the conclusion that once that we achieve a very deep molecular response and we keep it for a quite long period of time, the progression of the disease or the risk of progression is becoming less and less, almost disappearing with time. And for those patients that after discontinuation relapse, as we will see approximately, previously I showed a slide in which they were 60%, but the correct percentage at the moment, as you will see later also, is approximately 50%. Indeed, if they restarted the therapy, in most of the cases, they achieve again, almost all the cases, they achieve again not only MMR, major molecular remission, the safe haven, but also again deep molecular response. This is a very important concept that, of course, tell us that the worst that can happen for patients who are discontinuing the therapy is to have restarted the therapy that they have discontinued before. There is an interesting category of patients that are those that after stopping the therapy are showing again a reappearance of the PCR person. But if you do not restart immediately the therapy with TKI, indeed this initial increase is becoming a sort of fluctuation without overcoming the threshold of the major molecular response. And so from the clinical point of view, this is not relevant. The patient remain in a major molecular response and doesn't need to restart the therapy. This allows to increase the threshold to restart the therapy from the reappearance of the PCR Abelson transcript, so what we call MR4.5 or even lower, to the reappearance, to the loss of MMR. And indeed now the loss of MMR is the standard to restart the therapy. There has been a study, another study of the French group, recently updated at the ASH this year, showing that indeed patients that were discontinuing the therapy and were not losing MMR, in this trial, as you see, were 61%, so a percentage higher with respect to what reported in the STEAM trial. After 5 years, you may see that after 3 years, indeed, there are still some relapses. So the French investigators were curious to study better in a deeper way this phenomenon, and this is the long-term follow-up of these patients, in which after a consistent number of years, 45% of the patients are still in a deep and molecular remission. And what they have established, they have distinguished two types of recurrences. One is an immediate, which is occurring within the first 6 months, really, but some cases are also occurring during the first and the second year, or those who are late relapses of the disease between 2 and 6 years after the stop of the therapy. So this category, precise category of patients. And what they have seen is that those patients who are in a stable molecular remission in TFR without losing or really occasional increase of the BCR riboson, but which cannot be distinguished from this variation of fluctuation with respect to those who show a fluctuation more pronounced, which are represented in approximately 55%. These patients, this group here, are those who are at risk of losing later on MMR. And there are two messages that we can retrieve from this slide. The first one is those who are remaining for a certain period of time in a stable, deep molecular response, indeed are not losing. And probably they are entering the category of patients in which the disease, if studied with very sensitive methods, is progressively going down. The other category of patients with a more unstable deep molecular response, if you want, they are losing the response, but not in 100% of the cases, as you may see, but in approximately 1/3 of the cases only. And this is important. But what is important is that after a certain number of years, as again, as in the previous slide of the STEAM study, we can consider these patients out of the risk of the disease, of recurrence of the disease. And I would say that from the canonical, let's say, conventional point of view, in hematology, once the disease in the patients is not occurring again after 5 years, we can consider these patients really cured of the disease. And so, obvious question is the question that in part we didn't answer so far, which are the patients in which it's possible to try to stop the TKI therapy. There are recommendations. The most recent one is the one published by the European Leukemia Nest 2020 this year. As you may see, there are optimal in which is recommended at least to consider to stop the therapy. Duration of TKI therapy for more than 5 years, more than 3 years if MR4, and more than 2 years if MR4.5. And there are, however, other conditions which are mandatory, and in some other cases you can also consider to try to stop the therapy even if the conditions are not optimal, which is just because you need to stop the therapy for toxic effect, for pregnancy, for other reasons. Among these criteria, therefore, which is the most important, certainly is as you see in the results of this very important study called EuroSki, for the number of patients who were studied, that were approximately 800. As you may see, it's important to have a quite long treatment with TKI. This is statistically significant. But what is important, is more important, is the duration of the deep molecular response while receiving TKRs. And you may see that here is highly statistically significant, whereas the time of TKI treatment before TMR is not really important. So it's not statistically significant. So what does it mean? It means that we need to reach DMR as soon as possible and to keep it as long as possible. So the message is very simple. How many patients are falling in this category of patients who are reaching deep molecular response in for a quite period of time? And we know that if we are using in really matter, and these are data from the nested trial, which has been recently updated, will be published soon again after a 10 years of follow-up. 1/3 of the patient is achieving this goal if treated with imatinib after 5 years. More rapid is the achievement of a deep molecular response if we are using a more potent TKI, like a second generation TKIs. Therefore, if we consider, I would say, a scheme just to explain this phenomenon, we may say that we are using imatinib, which is absolutely, let's say, the king, the basic drug, if you want. We are remaining with some amount of residual BCR-ABL, which is due to the persistence of some subclones which are less sensitive to imatinib. If we are using a more potent TKI, like a second generation TKI, indeed we are in part overcoming the resistance of the subclones. Therefore, we are remaining with a lower amount of BCR-ABL. How do we know that the decrease of BCR-ABL is strictly correspondent also to the rapidity of the response? So there are two concepts, two sides of the same coin. If we are the persons who are responding very fast to the therapy, indeed, I heard previously Giorà, who said that he was already in deep molecular response after 6 months only after starting with imatinib. Indeed, these are those who are major chances to achieve later on very deep molecular response, and therefore the possibility to try to stop therapy with a high percentage of success. This is the optimal response independently from the type of TKI that we want to use. That is the minimum that we want to have if we want to achieve the possibility to try to stop TKI. BCR-ABL less than 10% after 3 months, BCR-ABL less than 1% after 6 months, MMR within 12 months, and MR4 within 24 months. If we are achieving these goals, it's very likely that you will achieve later on a MR 4.5, and after a certain period of years, you can try to stop the therapy. How many are these patients after 5 years of TKI therapy? This is, I would say, not, I would say, these are a scheme which has a deduction from the Neston study and published reported by Andreas Okaus et al. In 2015. If you are using imatinib, as you see, 1/5 of the patients, 21.6% after 5 years. If you are increasing using nilotinib, 300 mg twice a day, or another second generation TKI, we are increasing this percentage of patients. And there are 2 studies that I would like to show to you, which are telling us, however, again, which is the most important goal that we must achieve to try to stop the therapy. One is the nested triton. These are patients who were indeed treated first line with nilotinib for at least 2 years. They were achieving MR 4.5. They were maintaining MR 4.5 for at least 1 year of additional therapy, and if this was demonstrated with a very stable deep molecular response, they were allowed to stop the therapy, restarting the therapy only after the loss of MMR. And there is another study, which is extremely important too, it's called an nested stop, in which patients were initially treated with imatinib. They were responding well, but not sufficiently well to try to stop the therapy, which means that they were in between 1% and 0.1% of BCR-ABL. We're not achieving MR4. They were switched then to nilotinib, the second generation TKI, and they, after achieving MR 4.5 on nilotinib, they were indeed entered the consolidation phase of 1 year, continuing nilotinib, and then if this stable molecular response, deep molecular response was achieved, they were allowed to stop the therapy. As you may see, the curves of discontinuation are always the same. So we are always, all the curves that I will show you today are showing that there is a 50% probability of remaining in TFR, approximately, after stopping the TKI therapy. which means that independently from the type of therapy that you have started with. The only difference is not the time of the MR 4.5 to the beginning of the study, not the duration of the nilotinib treatment prior to TFR, but the total duration of the TKI therapy. So what does it mean? It means that probably what is really important is to achieve a very deep molecular response and to keep it for a certain period of time that, as I told you, can be at least 2 years, but maybe a little bit longer if you want to have more chances to achieve probably TFR. However, of course, to accelerate this process is good, but it exposes also the patients to the risk of a major toxicity. That's why nilotinib, as you know, is associated with a higher increased risk of cardiovascular events, which is not good in particular for elderly patients or for patients who have already the risk of cardiovascular events or developing cardiovascular events. So it's something that should be discussed with your doctor. I think that, however, fortunately, in the future, we will have other possibility. And one of the possibility is just to have a new category of inhibitor that we will see recently in the therapy of CML, which are different from all the other inhibitors, which are inhibiting, as the original imatinib, but all the subsequent TKI are not inhibiting the ATP binding pocket, but in this case, we are inhibiting the methylation site. Methylation site is something which blocks the activity of BCR-ABL. Of course, you can understand that the mutations that can occur can cause resistance to traditional, let's say, TKIs are different from the mutations which can interfere with this other process. And this may say that this is a drug which probably will be very useful to complement the activity of the other TKI. For the moment, it has been tested only as a third-line therapy. Before there were data on the phase 1 study just to establish the dosage. And indeed, it was demonstrated that even at the dosage of 40 milligram twice a day, of 60 milligram twice a day, indeed, it was in patients with a very old story of CML, it was able to demonstrate a very high percentage of even of molecular responses, and even in patients with the 359 mutation. But the most important aspect that these responses were associated with a low degree of toxicity, particularly cardiovascular level. This is something which can be very, very important to be considered in patients. Soon, as I told you, this drug will enter is a new TKI therapy it's not called a TKI, really it's called a stamp, which means specifically targeting the Abelson methylation pocket. However, this stamp drug will enter the market because in a trial in which was compared the third-line therapy with bosutinib in patients without the 359 mutation, indeed with the primary endpoint, very ambitious MMR at 24 weeks, indeed it was demonstrated to achieve this primary endpoint, and therefore this will probably be associated with the registration of the drug as third-line therapy. However, we are very curious to see the results of another trial, which is adding asciminib, as I told you, which is an optimal drug from the conceptual point of view to be added to the traditional TKI because they are inhibiting two different sides of the Abelson with imatinib with respect to those patients who are remaining on imatinib or those patients which are switching to nilotinib. This is just to see if in patients starting with imatinib, which is certainly most likely, I would say, the most diffuse drug for CML therapy around the world. And now in the vast majority of the countries is indeed quite cheap because it has become generic, are not achieving the goal of a deep molecular response using this drug. Indeed, imatinib only, indeed, but the addition of Abelson 001 will be able to improve the achievement of the deep molecular response, and therefore to try to allow. Are we ready to introduce TFR in clinical practice? I will finish with these few slides saying that indeed we are already doing this. In Italy, we recently published this paper, which was an observational trial on more than 300 patients who discontinued the therapy. I will go very fast now. All of them were in deep molecular response, one-third in MR4 at least, and two-thirds in more than MR4, MR4.5, MR5, or even lower. And the results are exactly similar, as I told you, to what we have seen in the traditional study. There were no progression, no risk of disease, and some of these patients are continuing in TFR. Those who restarted the therapy achieved again at least MMR, but most of them are again in a very deep molecular response. Again, this is what happened. I don't have time to go deep. Similar data have been recently reported also by the Spanish group, by Hernandez Boluda. As you may see, the percentage here of those patients achieving a successful TFR is even higher with respect to what in Italy. And the most important aspect of the paper published by the Spanish group is that indeed after discontinuation, there was an increase in the hemoglobin, an increase in the leukocyte number, the lymphocytes too, the platelets too, the creatinine decrease. So there is an advantage just to stop the therapy in terms certainly of quality of life, and maybe I would like you to discuss this point later on, but also in terms of safety. There is a withdrawal syndrome, but generally this is not so terrible, and it is lasting only for a certain period of time. Thank you very much for your attention.
Thank you very much, Professor Salio. It's always nice to hear you, even though it's not always easy for everyone to understand. I'll try to move along, and we have now some time for question and answer. And I just want to relate to the topic of TFR as we see it in CML Advocate Network. Of course, trying to start treatment is important, and it can be a goal of treatment for every patient when he works with his doctor, when they consult. But when we look at reality, and we saw this reality in the survey, the study we did among CML patients in 68 countries, we saw that because of the lack of monitoring in many countries around the world, and you cannot stop treatment today if you don't have access to a reliable monthly PCR testing. And that's mainly available in countries like Western Europe, North America, Australia, Japan, Korea, but in most of the world, South Africa, in Africa, in South America, in Asia, in most of these countries, patients don't have access to such reliable PCR, so they don't have the ability even to try to stop. So TFR is important, but for us, as I said, cure is much more important, because if we calculate how many patients are stopping today, even in the trials that are running, we see about 25% or 30% of the patient can stop. But that relates only to the patient in the Western countries. If we take the whole CML population, we are talking less than 10% of the CML patient. And that's not enough. As I say, for the CML community patient, we really want to try to achieve cure. And that's why CML Advocate Network is planning to do next month in October a scientific community advisory board with about 8 scientists who are working on different approaches to try to find cure for CML, and together with about 20 patient advocates from all around the world, we'll sit for a few hours, of course, on Zoom this time, and try to explore and see how we, the patient community, can support the investigator to drive forward the approach to find the cure, and hopefully we'll achieve that. And if I already there, and we move now to Q&As, and I apologize on behalf of Professor Pianani, who has another webinar coming at 7:30. She'll have to leave us even if we continue for a few more minutes. So on her behalf, I apologize. So the question is, and to all 3 of you, CML is one of the diseases we know molecularly best from all the other cancers. It only has 1 mutation, and that's why it was maybe easier to find the first targeted therapy to treat the disease. What makes it so complicated to eradicate the disease? What makes it so complicated to eradicate the stem cells, the leukemic stem cells that are existing? Why is it so difficult to continue to the direction of a cure? Maybe we start with you, Dr. Drucker.
Yeah, great question. If I knew the answer to that, I would get that into clinic as quickly as we could. The reality is that if you go back to some of the things I said earlier, it probably has to do with CML isn't perhaps as easy as we think it is. We certainly know the growth of the leukemia cells is driven by BCR-ABL, but perhaps the survival is dependent on other factors that preceded BCR-ABL. So maybe it's not just a 1 gene abnormality. Maybe there really are other things, or maybe we even have some data from my laboratory that if you shut down the enzymatic activity of BCR-ABL, the stem cells still can survive. So maybe there's more to BCR-ABL than just the enzyme activity. So to me, it's a little bit more complicated, and it's also complicated because trying to isolate these cells from a patient in a deep molecular remission when it's a very small fraction of their stem cell population, and trying to study them and comparing to the normal stem cells is a really hard scientific problem, but a critically important problem as you described.
Okay, thank you. Anybody wants to add anything? Professor Salio or Professor Anani?
Pia, please. I will after you.
I don't think after what Brian said I have much to add. I just remember again the late Professor Goldman talking about the dormant compartment, that maybe TKIs do not reach it. So might be that still maybe the only real curative treatment would be bone marrow transplantation, but of course we don't want to pay the price. So, and he also, again, I can only quote him, he used to talk about operational cure. So maybe we can live in peace with this small amount of cells with TKIs or without TKIs. But Beppe, I'd like to hear you.
No, I think that you're perfectly right. I think that the eradication is difficult, but certainly not impossible. The only system that we have to eradicate the leukemic stem cells, so let's call like this, is just represented by bone marrow transplantation, thereby allo bone marrow transplantation, and therefore by the possibility of immunological control of the residual leukemic cells. The problem is that using TKI, what we are doing, we are masking in part we are killing certain progenitors of the leukemic stem cells, but on the same time, we are indeed masking the stem cells because we are inhibiting the BCR-ABL. So we are making them very similar to a normal stem cells. And therefore, this is something that probably will require also not only TKI therapy, but something in addition. This is something similar to what Brian was previously saying, that probably we needed to discover something which is preceding or facilitating or I would say changing this CMLs themselves in a way to be recognizable by the immunological system again. So the phenomenon of the eradication of the leukemic stem cells is still a big, I would say, something that certainly it needs more exploration, more is a challenge for all of us, because if we understand these concepts, probably we will be able to cure not only CML, but most, at least, all the myeloid leukemias.
Thank you all. We have here in the, I don't want to start reading all the applauses and all the thank you that you have, speakers have received, and especially for you, Dr. Drucker, for the life-saving of so many patients and the participants here. But hopefully I can gather them and send it to you by mail later. We have a parent to a child which was diagnosed with CML, I think a couple of weeks ago. And he's asking if you can say a few words about the treating children with CML. Is there a difference between treating an adult or treating a child with CML? Do you want to take the question or?
Okay, I think that of course the disease in children is particularly challenging, because we need not only for I would say, because we are facing the same difficulties that we are facing in adult patients, but also because with the TKI therapy, we are in part interfering also with the growth of these children. So we must be very careful. However, they respond to the same drug. We are using, in most cases, imatinib, and there are also trials in which we are able to achieve at least a control of the disease after a certain number of years using after TKI therapy, using interferon in combination with TKI, and also in part some successful discontinuation has been achieved. So this is a little bit more challenging because certainly we have to deal also with the problem of the toxicity, which can be more important for children with respect to what we have for adult patients. Therefore, we do not want to use very, very potent TKI with a high degree of toxicities, because I think that this could, I would say, in a certain sense, change a little bit the perspective of their developments. So we must be very careful. But there are many trials now ongoing which can answer this question, and the survival of the children patients is good in our days too.
Okay, thank you. Pia, do you want to say anything?
Yeah, I want to say that I think we can take it even to a broader range. I think that when we think about patients, we should think about what is the purpose of the treatment, and treatment should be age adjusted. Just if we look at the opposite side, for example, the purpose for an elderly patient are different than those for middle age or young patients. So I think first of all, we should think what are the aims of the treatment. And for a child, it's, as we mentioned, not to disturb growth, thinking about the future, stopping, the option of stopping. And yes, the child is not just a small old patient. It has its own right.
Okay, thank you. I have a question of mine to you, Brian. Last time I saw you, I think it was a couple of years ago at ASH, I think was it New Orleans, if I'm not mistaken, where you presented the plan of Beat AML, and the plan that you are participating, trying to find solutions for the more difficult leukemias, acute myeloid leukemia, and there's really a very big plan trying. Do you think that the progress that you will be making with treatments for AML, will that be also suitable for blast crisis myeloid CML patients?
Yes, so thank you. First of all, I want to make sure that people recognize that I still have a significant lab effort in CML, but as one of my colleagues said to me at a conference, CML was a stupid disease, and I didn't take that personally. He was really trying to say what you said earlier, that it's a much simpler disease. So I have put some lab effort into a much more complicated disease, AML. which is very similar, as you note, to CML blast crisis. And certainly many of the things that we're learning about the treatment of AML could be used to treat blast crisis CML. But fortunately, blast crisis CML is a pretty rare occurrence these days. And Beppe and Pia, I wonder how often you're seeing it in your clinics, but I see it pretty rarely, and that's quite fortunate because it was an extremely difficult leukemia to treat.
Okay, thank you. I think the next question may be for you, Professor Salio. It's an Israeli CML patient, 41-year-old. She lives in the US, in Florida. And she said it took her 22 months to achieve 0.099 PCR, which is less than optimal response. It's like basically MMR after almost 2 years. And she was asking, would you recommend to try TFR if in the future she is able to maintain deep molecular response for at least 3 years, even though the speed of the decline of the PCR was slower, not optimal?
I think that certainly, but I would say if she maintains a very good deep molecular response for at least 2, maybe better 3 years, I think that it's possible to try to stop the therapy in this specific case. Of course, there are always patients who are responding very slowly, but this takes more time. But what is more important is the length of the deep molecular response that you achieve. So if then in a few, let's say, 1 or 2 years, we will have the possibility to use of one of the new possibilities that we are developing now to achieve a deeper molecular response, which means maybe a Cml or another drug, this is another possibility. So I'm sure that most of the patients that at the moment, you are perfectly right when you are saying that we are discussing about Western countries, most of the patients do not have the possibility of monitoring the disease and so on. But I'm sure that even if this represents only a small proportion of the number of the CML patients in the world, however, the experience that we are doing will, at a certain point, allows us to extend the possibility of TFR. I hope in a, let's say, a reasonable future, let's say 10 years, to most of the CML patients. So please resist. Be patient and resist.
Okay. I think we have some questions that were I would like to do some kind of a case study, a case story, and share with you the speakers. It's really a patient which is very close and dear to my heart. Her name is Michal. I have a daughter named Michal. And she was diagnosed with CML 3 years ago when she was 37 years in a routine blood test. Actually, she took imatinib, and even though it decreased in 3 months from 100% to 10% PCR, 3 months later, it already increased to 30%, so she was switched to dasatinib for 1 year, and the lowest she could have reached with dasatinib was 10%, but she had a lot of difficult side effects with dasatinib. At that point, her doctor started, she did the bone marrow biopsy and found she has a very, very complex karyotype, which I don't know what it is. She was put on 30 milligrams Iclusig, ponatinib, but after less than a week, she developed pancreatitis, and they decreased the Iclusig to 15 milligrams a day, and they started preparing her for a bone marrow transplant. 1 week before the transplant, she got pneumonia, and after 3 weeks with 15 only after 3 weeks with Iclusig, and her last PCR before the transplant went down to 2%. She had a very extensive chemotherapy protocol before the transplant, and then she did the bone marrow transplant. And she's now, I think, 9 months or 10 and a half months from the transplant. Because the PCR was still positive, And after the transplant, they put her back on Iclusig, and she's taking it with terrible side effects, really difficult. Even though she doesn't have difficult GVHD, the Iclusig is giving her a hell of a time, and she described it, my life is hell. I have very strong bone, joints, and muscle pain, and I am on 50 milligrams fentanyl and 24/7, and still in very pain. She cannot eat properly. I'm tired all day, but can't sleep properly. I am alive, but without a decent quality of life. She suggested the doctor to switch the ponatinib to bosutinib, but the doctor told her that he thinks the only drug that will work for her after all the difficulties and challenges she had is ponatinib, so he doesn't recommend to change. She suffers, she's hurting, she's on the edge of being desperate, and what can she do at this point of time? Is there any solution you can think about you would recommend to such a patient? And I know it's not easy to give recommendation over Zoom and over the internet when you don't know the patient and when you don't see them, but let's try to do your best.
But if I can say, is, oh, Brian, please, Brian. We cannot hear you.
You are muted.
I'll chime in after you.
Okay. No, I'm saying that certainly the story is, I would say, a very complicated story because of the transplant, because the resistance to at least 2 different TKIs with sensitivity only to Zik, which is associated with some toxicity and so on. However, the situation at the moment is from the disease point of view is good because the patients has responded very well to the inclusive therapy, and therefore for the moment I cannot remember exactly which was the response. Can you go down please a little bit?
You want to this question, to the one we asked?
Yes, yeah.
It's right here.
One, yes, sorry, Iclusig at the time is the look, and after transplant, 6 months with Iclusig, and the PCR is 0.07. So I would wait to change the therapy until we achieve at least MR4, which means 0.01 or less. And then we can also try to decrease the dose of Iclusig, maybe taking Iclusig every other day and so on. I wouldn't change the therapy because the disease is responding very well in cooperation with the graft versus leukemia effect of the transplant. But I would try little by little to monitor very strictly the response and therefore the PCR reimbursement, trying to decrease the dosage of the drug as much as possible without observing an increase of the disease. Then to switch for the moment, I think that the doctor is right. It's too early, not the best thing to do. Then we do not know the resistance to bosutinib and so on. So I would continue the same therapy, trying to decrease little by little the amount of the inclusive dosage.
Okay.
To try to improve the quality of life.
And that's a challenge. Any other comments, Brian?
Yeah. So I would just, first of all, I'm just so sorry that she's suffering so badly from this leukemia and from the treatments. I guess I'd start by looking at whether she's off all immunosuppressants, and if she isn't, it's certainly well past day 100, and it's time to begin to taper those, and that may actually help the immunological control of leukemia. And I also agree with Professor Salia that generally my approach with these leukemias are to try to get the most out of each medication that I can before I switch. So Iclusig is working. I would try to get to the lowest effective dose possible. If that means 15 milligrams every other day, or even doing what is probably not supposed to be done, which is to split the tablets and do 7.5 a day. Again, seeing if that would allow continued control of leukemia. But the reality is if somebody's post-transplant and is in a deep molecular response, it also gives you the ability to look at other options. So if Panat or Iclusig isn't tolerable at a low dose, but still has the ability to control, I would be okay trying other medications because you know you can always go back to Iclusig because it would work. As long as you're well below that 0.1 major molecular, I feel comfortable trying other treatments. But I say I do prefer to get the most out of each medication before I switch, because then I'm not switching back and forth, going from one medicine to another medicine, and then not knowing whether that's going to be the best approach.
Okay, thanks a lot. I think we have quite many questions, but I'll try to at least to put two more. One of them is interesting. It's another young CML patient from Israel who has been on nilotinib, and he changed to the dasatinib for suboptimal response. And after 3 years on the dasatinib, he reached 0.015% PCR. He wanted to see if he can stop. His doctor did not recommend him to do so, and he decided not to listen to his doctor, and he did stop treatment. And strangely, without treatment, he became PCR undetectable, 0. And this holds now for more than a year, the undetectable. So he's asking actually, did you see such cases where the PCR goes down to 0 without treatment and holds for long? And what is your experience with that? Brian, would you like to start?
I think Professor Salyer showed some examples where people who are off therapies, PCRs continue to improve. So congratulations. Good job convincing your doctor to stop treatment. And I hope that there's every reason to believe that at this far off treatment, as Professor Salyer showed, that this should continue.
Yes, absolutely. And I would add that we have also some cases who stopped the therapy because of pregnancy when they were only in MMR. Of course, we were waiting for loss of MMR to start any kind of treatment during pregnancy, like interferon and so on. But indeed, they were in the same situation that which has been described by these patients. So I think that there are still many mysterious things concerning the possibility to overcome the resistance of the residual leukemic cells and to achieve TFR and maybe a complete cure of the disease.
Okay. Before I go to the last question of the pregnancy, we have a patient. I think he's oldest with the most many years in our group. He's almost 29 years CML patient. He was almost 9 years on interferon, and then in 2000, of course, he was switched to imatinib, and he's doing well in regards to response, even though he had to go up to 600 milligram imatinib. So he's taking a higher dose of imatinib than the regular 400 milligram, but it's keeping his response in an acceptable level. But his creatinine has started to climb, and it has reached now 1.65, and he's worried. Would you recommend to change imatinib after so many years? And if you change, to what treatment would you recommend to change?
Please, Brian, to you, up to you.
I would do able sequencing, see whether there's a mutation, and that would help guide my decision. But with the numbers going up, it sounds like it's time to make some changes.
No, not the numbers. The PCR is not going up. The creatinine, the sugar, the creatinine level is going up, and it reached 1.65. 1.65.
Yeah, and so what's the PCR doing?
It's normal. I think it's around less than MMR.
Yeah, you know, one of the things that again, for people who are in MMR, you have such an incredible luxury of doing things. So for example, oftentimes in a patient who's is MMR or less, and I'm seeing some side effects, I'll give them what I call a treatment holiday. I'll stop treatment for 4 to 6 weeks, see what their PCR is doing off treatment, see what his creatinine does off treatment. If the PCR goes up and the creatinine goes down, I'd switch to a different drug. If the PCR stays down, I just continue to extend the holiday.
Yes, absolutely.
Okay.
I agree with Brian.
Okay, sounds a good solution to me for him with so many years with CML. And I think we'll move to the last question, and that's about the female. She was 31 years old when she was diagnosed with CML. She was 2 years on Busulib because she joined the clinical trial of Busulib. So she received Busulib for first line, and she reached MMR. After 2 more years, she has reached MR4. And she had 1 son before she was diagnosed with CML, but she desperately wanted another child. So together with the doctor, they tried to stop treatment and see if she can conceive. She lost her MMR after 5 months from stopping, and she had to restart Busulib. And she regained MMR within 2 months, and 3 months later, she regained MR4. So how does she continue now from here? She is 37 years old now and eager for a second child. What are her options?
Personally, what I think that for the moment, MMR the second time is very, very short. So I would continue for at least some other time taking Busulib, just one additional year at least, better 2, but I would say 1 additional year or 2 could be the right solution. Then I wouldn't stop the therapy trying to conceive, but I would try to conceive, to be very careful if there is a pregnancy ongoing immediately after, let's say, 15 days or so, just to be very careful about the timing, because at this time, if there is suspicion, even suspicion, that there is a pregnancy ongoing, is better to discontinue the TKI. During the first days of the conception, there is no mixture before the formation of the placenta. There is no mixture between the blood, the maternal and the fetal blood, and therefore the embryo blood in this case. And therefore, I would say this is not the dangerous period. The most dangerous period is that starting after the 3rd or 4th week of gestation. And so in this way, as we have already a proof that she was able to maintain MMR for at least 5 months, we will probably be able to get, I would say, the period sufficient to restart the therapy after delivery, so without the need to restart the therapy. So I think that in this can be, but this needs to be done eventually when there is a longer period of reachievement of the MR4.
Okay.
Yeah, I would completely agree with Professor Salio that, again, first of all, longer time in MMR, MR 4.5. I also completely agree that I wouldn't discontinue therapy while trying to conceive, but I would be extremely careful that the day you think you might have conceived, do a pregnancy test. If it's positive, stop therapy immediately. But the last point I'd make is that, remember, Zavi Zero Club was a complete cytogenetic response. That was a PCR value of 1%.
1%.
Let's not forget that a PCR value of 1 is still really well-controlled leukemia with a very low risk of relapse. So during pregnancy, I don't feel the need to restart therapy until that PCR gets somewhere above 1. And we know during the 3rd trimester that at least imatinib can be used with some degree of safety during the 3rd trimester. So 5 months to get from off therapy gets you to MMR. That's probably another 2 or 3 months to get a PCR value of 1. So again, I think you might be able to get through pregnancy after a little bit longer in a deep molecular response.
Absolutely, I agree.
Okay, thank you both very, very much. I really want to thank you, each one and one of you again. Professor Salio, for being again with us. Thank you very much for your great presentation and answers. And of course to you, Dr. Brian Drucker, thank you very much for joining us. I hope this can become a habit that we'll get to hear you now that I know that traveling is an issue for you with children at home, and you know how much I try to bring you to CML Horizon, but it was always difficult for you to travel, and we understand and appreciate it. But maybe now with the Zoom and the online platform, we can get meet together more often, and you can share your vast knowledge and experience with the CML patient community. So thank you again all. Thank you for everybody who has joined us. It was a pleasure having you, and I hope you enjoyed the webinar. It was really, really exciting for me. So I wish everyone good morning for you, or good evening for the others, or good night for the one from the East. And hope to see you very much. Have a wonderful World CML Day. Thank you very much.
Thank you, Giorra, for involving us in this beautiful webinar. Thank you very much.
Thank you.
Thank you all.